Hello, everybody. Welcome. Good afternoon to you from sunny Stockholm. It's a pleasure to be with you all this afternoon. Just before we get started to this really interesting webinar that I'm really looking forward to myself, can we check that all the technical things are working as we get you all into the room? So to do that, if you just go into the chat and pop a little message in there for me, let me know where you are from, where you're joining us from today, so that we can see that the chat's working, because this is where you're going to be able to ask your questions. It's also nice to get a little sense of who we're talking to today. Also, let me know in the chat if there's any technical problems. If you can't see anything, don't hear anything, any problems, that's for now. And also throughout the webinar, just let me know. So while, there we go, I can see Eva from Hammarby Stockholm, fantastic. And while waiting for more messages, let me just give you a brief introduction to Ebba Biotech. So we're hosting these webinars to really showcase some of the great science that is being done with our products. So we're a Swedish company. We were founded in 2015. Based on interdisciplinary collaboration. The products, the company, everything we do is based on collaborations from multiple different scientific disciplines, and I think that's going to be exemplified very well in our talk today. Our products are the Optotracers, and these are fluorescent tracer molecules for labeling macromolecular components. So that's who we are. Our speaker today is Dr. Susanne Löffler. She's a team leader at the Center for the Advancement of Integrated Medical and Engineering Sciences, or AIMS, and that is also based here in Stockholm, Sweden. And Dr. Löffler specialises in organic bioelectronics for bacterial infection. Her academic background has led her to publish very broadly in biomedical science and signal processing, and also she is a co-inventor of on the patents behind our products, the Optotracers, which is very cool. So I don't think you could really be hearing from anyone with a better, deeper understanding of the background of our products today than Dr. Löffler. So with that, I would like to thank you for your attention and hand over now to you. Away you go.
Yeah, thanks, Ben. Thanks for the nice introduction. And as you say, this was really like the aim, I think, of this webinar when the team at Ebba approached me to kind of help the customers to get a little bit of broader understanding of where the Optotracer technology is coming from and what has it been used for in like the very early stages of the development pre-commercialization. And that is really now what I'm trying to do with my presentation about opto-electronically active materials for infection detection and control. And this is really what we do in AIMS. And in terms of disclosure, I want to mention, as Ben said, and I'm a co-inventor on the IP relevant to this presentation. This is owned by Richter Life Science Development. I'm also active in Ebba as a product manager, and I'm also shareholder and member of the board in Fusifer, who is co-owner of Ebba Biotech. And the problem that we are addressing at AIMS is really biofilm. And biofilm are those bacterial colonies that are embedded in a self-produced extracellular matrix. So it's the ECM, the polymers that the bacteria produce, and the bacteria. And we want to detect both, but not necessarily as one big unit, but we want to be able to detect the ECM separately and the bacteria separately, if this is possible. And this hasn't been so easy so far. And this is also where the Optotracers come in. So the problem with the biofilm is also that it's everywhere. And we're looking for a simple, fast, and accurate methods to detect the bacterial growth, as I mentioned, and identify the bacterial extracellular matrix separately. And then we want to do this in liquid media, like in culture medium, or even body fluids, like in a later stage. And in the best case, we want to be able to do it on abiotic surfaces, like devices, implants, or catheters, and biotic surfaces like the tissue. because it has been notoriously difficult to detect biofilm in tissues. So, and our answer to this at AIMS are optoelectronically active oligomers and polymers. And first, of course, I want to talk about the optically active oligomers, which we at AIMS termed Optotracers. And these are molecules, small molecules with a defined length. They have side chains with defined position and charge, and their backbone is composed of ring structures. And interestingly, the central unit we can swap out and produce optotracers with different fluorescent optical properties and binding properties. And so the optotracers are fluorescent molecules that target biopolymers, and the signal is quantified by spectroscopy or microscopy. And when we talk about spectroscopy, where we acquire fluorescent excitation and emission spectrum, we can see here, like the high energy spectrum is the excitation spectrum, like the dotted line is unbound state, the continuous line is the bound state. And on the lower energy, we have, of course, the emission spectrum, which is redshifted, again, unbound is the dotted line and bound is the continuous line. And what we see is when it's bound, we have an increase in fluorescence. It's like an on switch. But what we can also see is when we compare the peaks here from unbound to bound, we have a spectral shift when the molecule is binding, so when it's changing its configuration. And that is a curve shift, a spectral shift that we also can use to quantify the signal of the optotracer. So we have like a bimodular detection method, like we can detect the intensity, or we can detect the curve shift, like the location of the peak. And this is best done with the spectroscopy, because in microscopy, we don't have this high spectral resolution often, but we can really use any fluorescence microscope. And this is really also the advantage of those molecules. We have, I have here like an example of confocal microscopy, but we can easily also use just the epifluorescence microscopy and even use the automated methods. And one striking advantage, I think, of the optotracers is that we can use it also with a super resolution microscopy. We have here like a Staphylococcus that we imaged with the Airiscan detector. And then we have on the other side like a Staphylococcus where you also see the septum, which we imaged with a STAT super resolution microscopy. And since the molecules are so small, you can really get this fine detail compared to what you have when you use antibodies in the super resolution microscopy. So in the first application really that I wanted to talk about, we are using Optotracers for detection of bacterial ECM components. So this is now only, we use the Optotracers to detect the bacterial extracellular matrix and not the bacteria. And here we're using Salmonella or E.Coli biofilms. And the extracellular matrix of those gram-negative bacteria are composed of cellulose and also curline. And curline is an amyloid protein aggregate with repetitive arrangement of beta sheets, as you might know, and cellulose is a carbohydrate made up from repetitive beta-1,4 linked glucose units. So you might ask, what's the common motive in those? And the common motive here is really the repetitiveness. So what we can say now is that the optotracers bind promiscuously to repetitive motifs, but we can resolve a target-specific fingerprint, often with some optotracers, not with all. And which optotracer is able to do this, we really have to investigate this for each molecule separately. But I'm presenting here a paper that is one of the very early works by my colleague Ferdinand Chung, who is now also assistant professor at AIMS. And he's been using optotracers for detection of curly cellulose in Salmonella biofilms using this spectral fingerprint that I've been talking about. So what he did is he's been using the clinical strain of Salmonella enteritidis, and he used the wild type and isogenic mutants which are deficient in cellulose or E.Coli or both. And obviously the wild type is can produce cellulose and E.Coli. So what he sees then when he is taking, he's growing a biofilm and then he's resuspending the biofilm in PBS, I think it was, and then he's acquiring the spectrum. Is here the excitation spectrum that you see in this graph. And what you see is that you get the double peak at like 450 and 500 nanometer around approximately. And when you have the wild type, you have the double peak. And when you have the cellulose, the E.Coli knockout, or like the cellulose control, you see here only the peak at the, you see the peak here at the higher wavelengths, like the lower energy peak. And when you have also the E.Coli in the biofilm, you see two peaks, or you see two peaks here, also at the 450 nanometers, like the lower energy peak. And this is really how Ferdi then extracted the different peaks and quantified cellulose and E.Coli content in the growing Salmonella culture. And that is what he did in this image now, that is where the x-axis is the time, and he imaged this for, I think, 40-something hours. And like he labeled the bacteria also with GFP, so the bacteria as such are labeled. And then he detected the 450 and 500 nanometer peaks and assigned them to cellulose or E.Coli. And then he was able to track the growth, like the biofilm production of like the cellulose and E.Coli production in the extracellular matrix of the bacteria separately. And he saw that both components come up then in the late stationary phase of the bacterial growth. So that was a quite nice finding early on. And we've since elaborated a little bit on these findings. And when he uses then microscopy, he really sees that we have the green bacteria and the Optotracer labeled ECM. And then when he does the overlay, we really see that it's like the ECM is like a corona that is produced by the bacteria. And we can, I think, have it here a little bit in a higher resolution. So we really see that the ECM is surrounding the bacteria. So we imagine this really like a quite neat method to study the biofilm formation in live bacteria. And like as I said, Ferdi has continued his research on this, and what he did is he again uses the GFP producing Salmonella with the different mutants, and now he grows this biofilm on agar plates, like I think he used LB agar plates. And he has the Optotracer in the agar, so this is also new. So he's using a supplemented agar that is containing the Optotracer, and then the colonies grows on this supplemented agar. And it's like a morphotyping, like a objective morphotyping, because morphotyping, we all know it can be quite tedious. But now he's doing here in the wild type, he sees like, of course, the green bacteria. When it's green and red overlaid, obviously we have yellow. And when we have the extracellular matrix only, we have here red. And it looks like here that there's these channels forming, and that there's like the ECM is guiding the way for the bacterial growth, for the extension of the biofilm. And it really looks quite striking. And then he knocks out the cellulose, and what's happening, all these nice structures vanishing, and we don't have these channels anymore. We still have a kind of a layering of bacteria and ECM, but we don't have these channels anymore. And when we knock out both, obviously we don't have much biofilm at all, or we don't have, at least what we can say is we don't have labeling, Optotracer labeling. Maybe there are other biofilm components that we cannot see with this method. And what we also can see here, unfortunately, from this, he used a different molecule in this study than in the previous study. And what we can see here is we have the same, more or less the same lack of red signal when he is using the cellulose positive and Curli negative strain, as if we were knocking out both Curli and cellulose. And that really leads us to think that mostly this Optotracer that we're using here is labeling the Curli, or at least it's an affinity thing, that the affinity to Curli is so much higher that we don't have any labeling of the cellulose. Because also when we put the Optotracer in the agar directly, we will not have excess of Optotracer like we have before when we put the Optotracer in the medium. I think that's the biggest difference of the two methods. Yeah, and because Ferdi's images are so striking, I put also this video that he has also here in the publication, where you can really see when the biofilm is doing what where. It's quite nice. And when are the channels forming and where are the channels forming? And here this gives us now the opportunity to take out exactly like one little location and maybe take a spectrum and see what component is produced at which time, at which location. So there's definitely more work to be done in this. And another study, as I said before, we want also to do it in biotic liquids. And another study is by my colleague, Haris Antipas, who is now in Singapore, has been using the Optotracers in urine from patients that were that had this recurrent urinary tract infections. And then he also used healthy urine, funnily, from our people in the group. I think Ben at this time also contributed to this study in some way. And then he used also spiked urine, that obviously we spiked the urine with cellulose producing bacteria. That is here, this little blue circle, and here the wild type. The little blue circle is of course the cellulose mutant, and the wild type is the cellulose producing bacteria. And then he took he processed the urine a little bit by centrifugation so that we don't have the artifacts by the pH. And then he took all the spectrum and he threw it into a semi-automated analysis, which is called principal component analysis. And then he plotted this in 2 dimensions. And what we really see is that we have some urines that cluster around the cellulose producing bacteria, and then we have some urines that cluster around the cellulose negative bacteria. So that means like 15% of those urines produce cellulose, of those urines have biofilm in the urine. So this is a strong indicator for us that urinary tract infections, recurrent urinary tract infections, might be related to biofilm formation in the urinary tract, and that this why we get this recurrent pathology. And yeah, so that would be great if we could like continue on that, but the problem is here that biofilm formation is not really yet a clinical symptom. And this is, I think, what Ebba Biotech wants to change in the future. Obviously not today, but hopefully in like 10 years. We want to bring the biofilm on the agenda, on the clinical agenda. Right, and then we switch over to the gram positives. Now I got a little bit talkative here. And the difference now between taking the gram negative, where we see not the bacteria, but the extracellular matrix, and then switching to the gram positives, is here in this project, we wanted to see the bacteria, we want to see the cell envelope. And the challenge here was to identify an Optotracer that binds to the cell wall of Staphylococci, but not like Salmonella and E. coli. And we have here two publications. That one is elaborating very much on the binding targets of this Optotracer that we then defined works best. And the other article is about what structural optimization we did to find the Optotracer really that works best for this method. And so Karen, obviously, she tried different Optotracers, and what I said also earlier, in the Optotracer we can switch out the central motif. And if we have a thiophene central motif, we see here like the purple line is the Staphylococci, the gray line is the Salmonella, and the dotted line here is the blank. And what we see here, when the central motif is thiophene, like the signal is lower than a blank, so we don't use it. When we switch out the central motif and use a phenylene unit, we have basically the same picture. But when we switch out the central motif and have a quinoxylene unit in the middle, then suddenly we get this striking signal fluorescence increase of like 10 times at least for Staphylococci, but not for Salmonella, and it's also much higher than the blank. So that is what we wanted to achieve. And that was quite striking for this group of more bulky central units. And of course, we did this now also with microscopy. And this is like fluorescence microscopy, like confocal overlaid with bright field. And that is why here we see the rods, the gram-negative rods, we see them here unlabeled, and in the cocci, gram-positive cocci, the S. aureus, we see them here like magenta fluorescence. And this is a mixed culture. So the bacteria have been grown in the mixed culture, or they have been grown overnight cultures, and then they've been mixed at different ratios. And that's what we can see here, like using the fluorescence, we can really track the ratio of Salmonella and S. aureus in the culture. And then we reproduce this also with another Staphylococcus and another gram-negative. So it seems to be like a gram-negative, gram-positive differentiation. And what we see here, what I say, also we can quantify the fluorescence intensity, but also we can look at the spectral shift. And this is what we see here. Look, the dotted line, which is the blank, and the Salmonella line, which is the gray. And then we have the like rosé line, which is the Staph epidermidis, and the purple line, which is the Staph aureus. And both bacterial lines, like staph lines, are shifted to the red a little bit compared to the Salmonella and the blank. And this shift happens in the excitation spectrum. And we think that the excitation spectrum indicates binding, and the emission spectrum indicates more things that happen in the environment, like osmolarity, pH, things like that. And then we did that also with different strains. What we did here is we plotted the optical density against the fluorescence intensity to see, is the bacterium growing when we add the Optotracers? So yes, the gray line here is the Salmonella, or the gray dots is the Salmonella, and it's really like then extrapolated and to give a flat line. So that means it's growing, the optical density is increasing, but not the fluorescence. So it's not binding, the Optotracer is not binding to the bacterium. But in the cases of Staph aureus and Staph epidermidis, we have binding, fluorescence intensity is increasing, and we have growth because the optical density is increasing. So this is how we can track binding and growth at the same time. And that helped us a lot when we were scanning now. Of course, we wanted to find out what is this Optotracer binding to. And then we were scanning a transposon mutagenesis library of about 2,000 strains, like it was a heap of plates that we were scanning. And this method helped us really a lot to do this process semi-automated at least. So we just put it in the fluorescence reader, we read the spectrum, we plotted RFU towards the OD 600, And then we had a script that was running and told us this strain is growing and the Optotracer is binding, or this strain is not growing and the Optotracer is not binding. And then we figured out that all the strains that the Optotracer was not binding to really had a deficiency in the cell envelope genes. And there is just a few. I don't want to go one on those, but it was deficiency related to lipoic acids, peptidoglycan, or the lipopolysaccharides. And that's why we went here with the purified components. We have here the peptidoglycan, where we can see, yes, we have a concentration dependent increase when we add the Optotracer. So we think it's binding. Same also with the LTA, with the lipoteichoic acids. We think it's binding the Optotracer to the purified component. Not so with the LPS. So we don't think the LPS is necessarily related, which also points towards that it's not directly binding the gram negatives because they also have LPS, obviously. Right. This was this. And then obviously we also did some studies with LTA knockouts, and those strains were provided by Dr. Friis in Copenhagen University. So we had a LTA knockout, and then we complemented the knockout again, and we see here that the knockout really gives this low slope, whereas the complemented strains and the wild type give the bigger slope in the curve. Right, and then we tried to, this was like the indication, okay, cell envelope is involved, but we really wanted to see that. So we did GFP as aureus, and then we labeled the Optotracer, and this is now, I think, the Airiscan microscopy, or I think this is normal confocal, but it's super pixelated because we reached the optical limit, and this is now with the Airiscan microscopy. We really see like the empty cell inside, we believe. And here, interestingly, it was not possible to do the GFP labeling because the GFP was bleaching, which the Optotracer didn't, which is also a strong argument for using Optotracer in these high intensity microscopy studies. Right, and now in the last minutes, I wanted to switch over a little bit to the rest of my work and bridge towards the active surfaces, which really chemically have a lot in common with Optotracers. And they are polymers, and they are doped with polymeric counterions, and they're redox active. So what we're doing with those, we make composite surfaces. We can couple things to this conductive polymer surface. And what we did here, we used this active opto-electronically active surface, and we coupled silver nanoparticles. That means we have A, the silver nanoparticle, and B, we have an electronically active surface, which we can constantly electronically address. And that we did here. So we applied a 5 Hz square wave to maximize the current flow. So we always constantly want to have current flowing to confuse the bacteria. And what we see here that is the PET control. We quantify the biofilm in this study with crystal violet, but I promise you the next study, we want to quantify the ECM with the Optotracers. So PET and just the conducting polymer gives approximately the same amount of biofilm. Interestingly, also the electronically enhanced, like where we apply this square wave, gives the same amount of biofilm. But when we add the silver nanoparticles, then we get a reduction in biofilm. And when we have the silver nanoparticle plus the addressing, we get much more reduction. And a follow-up study on this was where we just electrochemically oxidized or reduced this polymer that the bacteria were growing on. And what we've seen is that in the oxidized material, there's a lot of biofilm and not so much on the unswitched or reduced. So our theory is now that the bacteria use the oxidized material as a respiratory electron sink. And that is why they're growing so nicely on the oxidized surface. And that is obviously something that you can use for different kinds of applications. So we're going forward with that really in the next study. And at the same time, of course, we want to detect bacteria not only optically, but we also want to have an electronic signal, because electronically signals have the advantage that you can send them somewhere else. Like digital is always nice, and you don't have to process them like super much with AI methods to just get one data point. So what we did is we made this little 2-electrode device where we had also from this conducting polymer, we had also the ion conductive channel in between. And then we added different redox agents, and we saw that the sensor responds very nicely to those redox agents. And it is not so much related to how many redox groups they have, but it's more related to the size, like the sensor response. So, and then we grow bacteria on the sensor, and what we saw is in all the bacteria we tested, like from S. aureus to Klebsiella to Pseudomonas, we get a signal. And interestingly, we get the signal also when we ultrafiltrate the bacteria. So we get a signal that is most likely mediated by small metabolites. And that is the most interesting for us. And we later showed using all these electrochemical techniques that is really like oxidization of the material, and not, or like, no, not oxidization, so reduction of the material, and not like ion concentration related. So that was really nice. And then we did the same kind of study, because, you know, we want to see bacteria and we want to see biofilm, and we changed a little bit the device architecture and made it into a transistor. And that was maybe mostly Ono and Karen who did the work here. And we used the salmonella that did produce the biofilm, and then we used this biofilm deficient mutant. And then we did quantify the source drain current. So we were gating the transistor with like 0.5 volt. And then we measured the current that was flowing through the channel that is made up of this conducting polymer here. But interestingly, in both cases, we got like a growth curve. And that is also, again, telling us, yes, probably not the bacteria, not the extracellular matrix we're seeing. It's probably small metabolites that really make the signal. So yeah, next step of the work is really trying to find a like discriminator for bacteria and biofilm in this work here. And obviously we have ideas to use the Optotracers for that, but we haven't really gotten to like the pinpoint exactly how we are doing that. But it's work in progress, and it's work in progress by the fantastic teams in the Agneta Richter-Dahlfors group. We have our team leaders, Onur and Kira and Ferdinand now, and also myself, who has a very tiny part in this. And also our PhD students and master students who contribute a lot in the work, and of course our collaborators, mostly in Linköping, who are the chemists who exactly know every little configuration of the Optotracers, and Mikael Rehn, who's in Umeå now, and he knows everything about Salmonella, and obviously our Danish collaborators, Dorte Fless and Daniel Otzen, who help us a lot in figuring out what's going on with the biofilm. Yeah, right. Thanks for listening. That was fun.
Cool. Thank you very much indeed.